several years ago there was an article in one of the archaeology magazines that talked about some of the findings, older settlements in the south than there were in the north. Now that couldn't be if they're all coming from the north on down. How did they get to the south? I think more and more scientists are open to the possibility that at least small groups may have come across by, it would have to be most likely oceanic crossings, perhaps through the Polynesian areas and then eventually on to the New World. And those would have just contributed little bits of DNA and other populations to a large group that most likely was populated from the land bridge.
The fundamental question is, what is being sampled? It's been estimated that the population of the Western Hemisphere during pre-Columbian times had reached levels of 80 million inhabitants of North and South America. But then it has also been suggested that by the late 16th century, between 1500 and 1600, that population had plummeted to less than 10 million, from 80 million to 10 million, due to the exploitation of the native population by colonizing influences. And clearly that number continued to decline in the wake of Western expansion in North America and throughout the Western Hemisphere. So the point being that the population that is being sampled today is a mere fraction of the genetic diversity that existed just a few centuries ago. So whether it's random or not begs the question of what is being sampled, and does that take into account the genetic diversity that existed at one time, even if a sampling of that genetic diversity would reveal Book of Mormon peoples, their presence, their trace.
When Columbus in 1492 came to the Americas and made his first stops in basically the Caribbean islands and in Haiti and some of the islands around there, they estimated, or perhaps scholars since then have estimated, there was maybe in the neighborhood of 250,000 to 1 million people in those island areas. 50 years later, they figured there was about 500 people in those island areas, and 100 years after that, by about 1650, they believe they're basically extinct. Some scholars believe that up to 80% of natives were killed off by either wars or diseases brought by the Europeans. And that might be high. We don't know exactly. Like I said, it's difficult to say with a certainty, but that's what some scholars estimate. Either way, there was a lot of people wiped out, a lot of history that went along with it, and a lot of DNA that would have disappeared as well. And so you reach these bottlenecks and lucky genes that can move forward, and other strands of DNA would have died out.
Individually, our genetic makeup reflects only a fraction of the potential genetic diversity represented in our genealogy, in our ancestry, because, for example, our mitochondrial DNA is passed down maternally. So I received my mitochondrial DNA from my mother, who received it from her mother, who received it from her mother. So if you think of that, that 4-generation chart, mitochondrial DNA only traces that bottom line on the chart. The mitochondrial complement of all the other lineages there, on that chart, what is it, 16 on the right-hand side, 16 individuals. 15 of the 16 have no representation in my genotype. Likewise, my Y chromosome, which is the other principal target or locus of interest because of its strict paternal inheritance, traces the upper line on that genealogical chart and only represents contributions from that paternal ancestor at the very upper right-hand corner of the chart, one again out of 16. So all the rest of that is not represented, at least in my person. There may be other individuals, obviously, of my generation that have some representation, but they may not be sampled in the study, for example.
When my wife and I have 5 kids, each one of those children has 50% of their DNA from me and 50% from my wife, right? So already we've reduced our DNA data set by half, in these children, right? When they have children, their children, my grandchildren, will have a quarter of their DNA from me. So every time, in fact, every generation, you're diluting what was a single genome by half and half again. So, and then you throw on top of that, in a population context, the concept of genetic drift and fixation of variants or loss of variants.
What is the effect of infusing a very small colonizing group, very small gene pool, a literal drop in the bucket of the gene pool of the existing indigenous population of the Western Hemisphere? And would we even expect to find a genetic trace of that inserted population?
There have been many individuals well versed in this area who says that we can only catch maybe a glimpse and perhaps now and again a shadow of what these populations looked at in the past. In fact, literature will quote that up to 64% of ancient haplotypes are now extinct.
And there are multiple examples of contemporary populations where the genetics of their traditional origins, through oral history, through archaeology and so forth, do not corroborate that claim.
In Jerusalem at that time, there had already been major minglings between different groups, and so DNA was introduced from various parts of the world. So we don't have a standard to compare it by to say this is Lehite DNA.
The Middle Eastern area has historically been one of the most fought over areas of the world. There have been conquering armies that have come in and wiped out peoples, whole peoples, and flooded the area with DNA contributions from entirely different areas of the world. That has to have an effect upon the gene pool that's available.
Most of the Jews from Eastern Europe and Western Asia were converts, the Khazars and other ethnic groups that had nothing to do with the Israelitish bloodlines of that time. So you don't have A and you can't establish B, so how in the world can you possibly compare them to test your hypothesis? Your assumption that if there was an insertion of Lehi and his small colony, that there must be some marker like the Cohen marker in order to reveal that presence. It's a totally illogical argument.
The Cohen haplotype comes from a marker they presume was descended from Aaron, the Cohen priests, and they were forbidden to intermarry with most other Israelites, and so it was a very tight-knit group, and so this Cohen marker was passed down from generation to generation. Only about 2% of Jews have this Cohen marker, and the problem is that about 2 to 3% of non-Jews also have this Cohen marker. So there's a few of them out there. It's a strong evidence that somebody is descended from Israel through this particular lineage, but it's not proof because, like I said, there are non-Jews that also have this marker. The problem is that we find this marker in the Lemba of an African Bantu tribe, and they had traditions that they were descendants of Israelites, and they came to Africa and carried on traditions that were very similar to Jewish traditions.
But the point being that these Lemba on the surface couldn't be distinguished from the other indigenous peoples of Ethiopia. But they had this oral tradition of an origin from the Middle East. Well, it just happened that one particular clan out of 7, I believe, showed the presence of this Cohen gene marker. What if that clan had been ravaged by disease or hadn't been sampled in the study and only the other 6 had been sampled? There would have been absolutely no genetic evidence whatsoever to corroborate their oral tradition.
The problem is that that's not the same lineage that the Lehites came from. So there would not be a Cohen marker to examine it by. If we look at this African tribe without the Cohen marker and just examine DNA, standard DNA, mitochondrial DNA, they're no different than the other peoples around them. So if it wasn't for the Cohen marker, there'd be no reason to believe that the traditions are correct. And we won't expect to find a Cohen marker among the Lehites. If we did, then that might be a whole other story, but it's just not going to happen.
There are